Hello, everyone, and welcome back to the Bioinformatics series. This is the last course we'll be teaching, and I will be wrapping up some of what we were discussing on differential expression analysis. Like before, this is going to be a bit of an R-heavy class. I have some slides just to kind of introduce the topic, but it'll be mostly a walkthrough of some R code that should be modular enough that you can kind of cut and paste your own data into to generate analysis. As I mentioned, we'll be focusing on the visualization of DE-seq today, and that includes heat maps, volcano plots, Pca plots, and at the very end, I'm going to give a brief overview of how to do gene set enrichment analysis. So let's dive in. As before, I'm going to start us off from the very beginning so everyone can, so we're all on the same page. Here I am on the Bioinformatics 2020 homepage. And here for under lecture 19, there are slides available. I'm going to open those up and we're going to present those. If anyone wants to follow along, they can. On the very first page, you'll see a link to a Google Drive folder. I'd recommend you also open that because in there will be some of the code and data that we'll be using later. For now, I'm first just going to go through these slides quickly. So through the first lecture, we spent our time working through what differential expression is and how to run through some basic analysis using DE-seq2. Today, we're really going to focus on the visualization of the analysis. So the visualization is something that we'd wanted to spend a whole lecture on because in the end, your data is useless unless you can properly display what it actually means. And there's lots of different ways you can visualize this analysis depending on the message that you're trying to send. So as a recap, we did differential gene expression analysis before using DE-seq2, and it resulted in a table that looked something like this. In this table, we have the base mean, which is the average expression for that feature, for that gene. The log2 fold change, which is the difference between our A and B group. The LFC SE is the standard error of that log2 fold change. And then the statistic is the statistic for the statistical test. And then p-value and adjusted p-value are the respective statistics for this test. Up next, we're going to talk about visualization and gene set enrichment. The first visualization we're going to go over is a volcano plot. Volcano plots are a great tool to get the full results of your DE-seq analysis or any differential expression analysis in a single plot. They're effective because they show the two most important characteristics of your differential analysis, the p-value and the log2 fold change. And with a couple transformations, you get it so that the p-value increases with significance and the log2 fold change splits around the 0 for greater than and less than changes. Oftentimes, these points are then colored for emphasis. The next visualization we're going to go over is PCA. So PCA stands for Principal Component Analysis. A whole lecture could be devoted to PCA, but we're not going to do that. We're going to focus on the upshot of it. But basically what PCA allows you to do is reduce a multi-dimensional data set down to 2 dimensions so you can visualize how groups can separate and cluster. So this is a useful analysis for something like gene expression analysis because you have 20,000 features, but you can't look at all those features on a single graph. So this will take the orthogonal projections and allow you to put it into a 2-dimensional chart and then look for your subgroups. And the last visualization we're going to go over is the heat map. The heat map is a fundamental visualization in virtually any sequencing-based analysis. It's a way to visualize all your features and samples in a single figure. Heat maps can be really helpful. Heat maps can also be really ugly. And making a good heat map can take a lot of work and a lot of tinkering, but oftentimes they are a very effective figure 1 for any kind of analysis or any kind of project you have. So we're going to spend a little time on how to best do that in R. Lastly, we're going to go over some gene set enrichment analysis. I have a couple slides here on a basic way to do this using Broad's GSEA, which includes using MSigDB, which is their curated database of molecular signatures. We're going to do this at the end in the walkthrough, but I have left a couple slides here to kind of show what that is. Because again, this slide deck is mostly used for reference more than anything else. So that's kind of all I wanted to show as far as an intro. But like I said before, most of this will be actually coding, so let's switch over to that. Also in this folder, as I mentioned before, is code and data. The data has an additional file in here, which is another DESeq file. The differential expression output we had from our Pca data set last time is fine, but because I believe it's a Drosophila, I wanted to use a human gene data set to look at gene set enrichment analysis later. So we're not going to use this for a little bit, but I'm just going to download this for now so we have it ready to go. And then as far as the coding material goes, I have two more markdowns in here. I have 2 and 3. So we're going to download those and we're going to open them up. So starting with 2, this is the vignette I prepared for you to go over the visualizations of differential expression analysis. We have the 3 figures here that I was talking about before. This will be a lot of just R coding that I'll be demonstrating live in this window over here. I will try and slow down on the parts that I think are confusing. And there might be some snips in here that will be a little more complicated, but I just won't spend time on because they are, again, that kind of plug and play functionality, where as long as you change the parameters up top, then the rest of the code should respond accordingly. So we're going to start by loading in a couple of libraries, ggplot2 and DESeq2. DESeq2 we've seen before, which was used last time to actually do our analysis. ggplot2 is a very common R package used for plotting. We're going to load in all of our files and run the differential expression analysis again from last time. These paths are my paths on my computer, but you can put it wherever you are downloading the data files to. So I'm just going to run this whole section real quick just to get through the DESeq part of it, and then show that we have the same output that we have as last time. So now for visualization. Oh, one last thing. Last time I was asked about this, and we will need this as well later. DESeq will do the normalization of your counts. It's a part that's built into the algorithm. But you can also grab out the normalized counts using just this little bit here. So after you have created your DDS object, all you need to do is estimate the size factors, which is the function that will essentially do the calculation that will prepare for normalization. And then if you run this line here, counts on the DDS object are normalized true, you can grab out your normalized counts. So we're just going to run these 3 things here. So we're going to do that later. So let's start with the volcano plot. As we said before, we have our differential expression table here with all the various metrics. The 2 most important things though for visualization are log2 fold change and the adjusted p-value. So the first thing we're going to do is we're going to make a new table, or we're going to grab out these 2 columns. The next thing that we're going to do with this table that you can see down here is we are going to transform the adjusted p-value in a way, so as I alluded to before, that it will increase as it becomes more significant. This is just a more intuitive way to conceptualize these things. So by taking the log 10 of it, the negative log 10 of this value, you will then have a metric that will go up the more significant you go. And I'm going to call that p-stat. So now we have this table that looks like this. Using this new table, we're going to do one last thing, which is to clean up our table and to just get rid of some errors later. Any NA values in here, which I alluded to last time, that there could be in the adjusted p-value when the base mean is so low, the test doesn't even do anything. we're going to turn all those to a p-value of 1, which is basically an insignificant value, but it's just cleaner to work with when we're doing visualizations. So I'm going to turn all those values to 1. Now, making the actual volcano plot itself is pretty straightforward because a volcano plot, in its essence, is just a scatter plot. You have one value on the x-axis, which is the log2 fold change, and one value on the y-axis, which is now your pstat. So we're just going to use ggplot and make a scatter plot with these two values. A little review on ggplot, that ggplot is a fundamental package used for really any kind of plotting things you need to do in R. You initialize it by running this ggplot function and then giving whatever object it is you want to plot on, and then giving the aesthetics that you want it to use for the plotting. So in this case, we have our table here, vol plot tab, and we want to use on the x-axis log2 fold change, and on the y-axis pstat. So after you initialize the object and then tell it to run with geom_point, which tells it to do a scatter plot with this data, we'll then be able to generate the plot. I will be generating the plots here, as you can see, but just to keep things organized and so that you can see what's going on, I'm just going to kind of show you over here. So this is the plot that's generated from there. So this is a good start, but there's a lot of things that go into making a good volcano plot that make it a much more effective visualization. The first of which is really the main of it is the coloring. So you want to be able to emphasize points that are either significant by p-value and or log2 fold change. So here's a little bit of a large code block, but I promise it actually makes sense if you look into it, in which we're going to do that coloring. And as I mentioned before, a lot of these code blocks will have the only things you need to change at the very top, and then the rest will kind of adjust as is. So let's copy and paste this into the R window. So the p-value here, we're going to have a cutoff of 0.05, which is a pretty normal cutoff. We're going to have a log2 fold change of 1. Now, what the rest of these lines do is create a new column in our vol plot tab called color. And then based on these metrics, it's going to assign it different values, such as significant p-value up and significant log2 fold change up, both up, p-value down, log2 fold change down, both down. And it's going to use these metrics that you input at the top. So after you run this entire section, you'll be left with a volcano plot that looks like this. So now you have this column here, color, and you can see that in the color column, you have these different values. You have 7,000 genes that are not significant, 106 that are significant for both log2 fold change and p-value in a downward direction, and so on and so forth. So now with this new column, we can do the same ggplot call we did before, but add on this aesthetic of color. And by doing this, it will then color the dots based on whatever the indication is here. Now, where is it going to get those colors from? It will pick its own colors, but I think it's important to pick good-looking colors as you do this. So I actually made a little color vector here that essentially is going to line up each of these colors with these values. These may not look like colors, but they are. Basically, if you go into any kind of hex finder online, it'll allow you to pull out these 6-digit numbers that are indicative of colors. And this is a really good idea to find these specific colors, because in general, the color palette, if you just put in red and blue, is not very good. And it's a good idea to be really, really particular about what colors you pick. So I'm going to input this color param vector, so we have that to work with. And then we're going to regenerate this plot. And now it will look like this. So now we have the coloring done on our plot. So with just a couple more aesthetic tweaks, we can fix up the axes and have a nice looking volcano plot. There it is. So the last thing we want to do, and I wanted to include, would be how we can label these points on the plot. So there's a nice package here called ggRepel, which will basically allow you to attach labels onto your points of interest, and it will automatically push the labels away from the points as to not block them, hence the repel part of it. And what this block of code does is, using that library and using that function, we are going to grab 20 genes, and I'm going to grab the 20 genes that have the highest adjusted p-value. That's what this line does here. But you can have whatever genes you want, as long as you end up with a vector called labeled genes, which is what I have here. So now I have all these genes that I'm going to label, and now I'm going to make a table with those genes using this, where in our plot tab, I'm going to pull out from the row names of the Volcano Plot tab these labeled genes. So as you can see, here's label tab. And now using this, I can add this last block, which again, I understand this is one of the sections where it has a lot of code to it, and you can do a lot of tweaking. But as of now, this is the kind of thing you can just kind of plug and play into your own analysis, and it should be able to figure it out pretty well. And what this does is that it now attaches all these labels to these plots to emphasize which ones you want. You can see in the little window, it's going to look like nothing because it's too much room. But as you actually blow it up, the PDF will adjust to slide the labels into place. So that's pretty much how you make a volcano plot. This is a really useful figure to help visualize your DESeq2 output, because you can show all of your genes, you can emphasize the ones that are significant by whatever metric you decide, you can label genes of interest, and it's always a good place to start. The next thing I'm going to cover is Pca Plots. As I mentioned before, I'm not going to dig into too much what Pca actually is, because frankly, it's a lot of math and a lot of explanation that's not pertinent to this class. So instead, I'm really just going to show you how to visualize it and why it can be useful. So as before, I said you can grab your normalized counts, which we will need for this visualization using just this line, counts on the DDS object, and then normalized equals true. Now, before I create the Pca plot, I wanted to show that oftentimes for these kind of global analyses, you have your however many genes you have, which I think is something like 9,000, 8,000, whatever, 839. Now, you can use all this information to display these global visualizations, but in the end, it's much more useful to grab a subset of those that can exemplify the variance that is in your population. So here what I'm doing is I'm essentially just going to grab the top decile, the top 10% of varied genes. By doing this, this will help kind of clean up the data and what we're looking at, and it will also focus on the things that are really driving the differences in our population. So these lines here show that I'm grabbing the 90th percentile, or the top 10%. I'm going to sort all of our features by their variance, which this line does here. And then I'm going to select out the top decile of genes and create a table with that. So this is the dimensions of our new table. And as you recall, this is the dimensions of our old table. So we've selected for the top 10th of genes. Pca plotting takes a few more lines of code where to start with, your Pca data has to be a transformed version of the table because it just needs to have the samples and features on the opposite axes, just the way it goes. So that's the first thing we're going to do. We're going to transpose the top var table. That's to make the Pca data object, and then to actually get out the principal component analysis. So the values associated with the principal component analysis. We're just going to run this one function, prcomp. Now, prcomp is going to output this object, Pc data, which will have a bunch of things associated with it. Again, I'm not going to go into the specifics necessarily of what those things are. You can look those things up on your own, but I am going to talk a little bit about the parts that we actually need. Specifically, we want to focus on the X part of it and the rotation part of it. So the X part of it is the actual principal components themselves. So if I just grab this real quick, this is a 7x7 table for your 7 samples and then 7 principal components. And this is showing the value of each of these principal components for each of these samples. So I'm going to pull out the first 2 principal components because I will need those later for plotting. Again, I'm reducing down our entire population to a 2D axis. So I'll need the principal component 1 and 2 for that visualization. And then I'm pulling out what's called the loadings of the principal component data. So This is a useful thing that I like to do that some people leave off their principal component analysis, but I find it to be very useful and help explain the figure a little more. So what PC loadings are PC loadings are essentially how much each of these features you had actually contributed to the principal component itself. So you can see that when you do this type of thing, if you want to know, okay, well, what's really driving the spread of the on our principal component one axis, we can actually pull out the top genes, the genes with the top loadings for each of these principal components, and then carry those later to the visualization to show that here's all of our data reduced down to two spaces, principal one, principal component two, and now I'm also going to tell you what it is, the features that drove the variance on that principal component. So by doing this, and when you're actually explaining this and showing to someone else, you can help demonstrate what's really happening in the underlying data, as opposed to just kind of the hand-wavy explanation that principal component analysis oftentimes is. So using these loadings, I'm just going to grab three genes, the top three genes for both principal component one and principal component two. And then the last value I'm going to grab is the amount of significance explained by each principal component. And that's grabbed by the summary function. So summary of the PC data will return this little table here. And then by grabbing this value from each of these, you'll get the percent variance explained by that principal component. And I'm also going to use that in my visualization. So we pulled out a lot of information there, but it's all worth it in the end, because now what we get is we get a Pca plot, which again is at its essence just a scatter plot. So a lot of these things will seem familiar. ggplot with the data being our Pca plot data object, our x and y axes being the two principal components that we assigned here, and then the coloring coming from our annotation table. So after we have done those three things, you'll see that the Pca plot will look like this. And you'll see that this Pca plot, like all Pca plots, will be two principal components showing our samples in a two-dimensional space. But the addition of these extra things will help explain what's going on a little more. Specifically, by adding on the percents to these axes, you'll be able to explain how much variation is explained in each of these directions. And also, these three features are the primary drivers of this variation, just like these three features are the primary drivers of this variation. So in the end, what you have is a nice figure that shows differences between your groups, how much difference can be explained in this one figure, and then the primary features that help drive that separation between your clusters. So after PCA and Volcano Plot, I wanted to show just one more visualization, but I figure it's probably the most important and the most asked for, which is the heat map. So there are about 15 packages for heat maps in R, and it's not an exaggeration. It has been done to death, but a lot of people do their own thing. They've been included many times, and people have tweaked it in many ways. There's a lot of ways to do a heat map in R. I'm going to show you my favorite, which is also a very popular one, and the package is called Complex Heat Map. Now, the upside of this package is that it's heavily used and it's really good, which means you can literally do anything that you want with it. It has a very extensive vignette that's very well maintained that will explain all the things it can do. And overall, it just seems to be one of the best ones out there. Now, the cost of having this complexity is that it does take a little more coding and a little more reading to actually be able to use it. Now, you'll see that it's actually not that much in the end, but whatever coding it is well worth it to create a nice figure that helps demonstrate your data and is of publishable quality. So let's dive right in. We're just going to load up the package, and I just want to show you right off the bat that using our top variation table, you can create a heat map using a single line, which is just this heat map, and then this top variation table as a matrix. It has to be a matrix for this function to work. Now, what you'll see from here is that this is really not a nice heat map. There's lots of things that have to be done to actually annotate it and make it look good. But there's also one key thing missing from this, which is a fundamental part of creating a good heat map, which is making your features each comparable across samples locally without globally shifting the whole thing to make it unreadable. So what do I mean by that? I mean, Basically, your global scale of counts, as you can see here, is from 0 to 8,000. But the number of features that actually are up in this 8,000 range is teeny tiny compared to the rest of the population. So when you're trying to show all of these samples and all of these features at once, what you get is you get this scale centered around 4,000, which is going to make virtually everything seem blue. So the way you address this problem is by scaling each feature individually across all samples. So what this will do is, it's essentially just a z-score operation, but for those who are maybe a little unfamiliar with that, but basically what it'll do is that it'll look at the 7 samples across each feature. It'll set the average to 0. So if the average is actually 1,000, it'll subtract 1,000 from everything and it scale everything down. And then it'll just assign each value its standard deviations away from a mean of 0. So what this allows you to do is this time when we make our heat map, I'm going to make a heat map plot tab here where I do a couple of things here, which I will take the 10 seconds to explain because it's worth understanding. Where basically I am taking our top varied table, as before, which is our top 10% of genes, about 800 genes or so. I'm applying a function across every row. That's what this means. Apply across every row, one being rows. And the function I'm applying is the scale function, which is going to perform the operation I just alluded to, which is the z-score scaling. Once you do all that, all of this is going to transpose the table automatically. So we actually want to transpose it back. A little obnoxious, I know, but that's how it works. And then we're going to turn it into a matrix again. So after all of this, we will have a heat map table that looks like what we had before, the same dimensions and the same look, but now the data has been scaled. Note that this operation drops the column names, so I'm just going to throw the column names back on from before. Now this time when I plot the heat map, you'll see that it's a much more legible table, that everything has been kind of brought down to scale. And you can already tell from the axis that these samples are clustering by a treated and untreated, which is a good indication that there is some differences happening here. Now, when you look at this heat map though, I mean, maybe this is kind of a treated block and this is kind of an untreated block, but overall, it still looks a bit of a mess. Now, the reason it looks a bit of a mess is because you're still putting in all 800 of your top varied features. Now, top varied features doesn't actually mean top difference between your A and B groups. Some of those features will be inherent because the differences between the group will lead to a higher variance, but there can also be some features that are highly varied at a basal level across all samples, which will also sneak in here. So what we're going to do to clean up this heat map, oh, we're actually not going to do that quite yet. That's another section down. Sorry. The first thing we're going to do is we're going to add an annotation onto this. So what this section is, I apologize for going out of order. I forgot we did this first. Doesn't matter. What this section is a fundamental part to making good heat maps is to annotate them correctly, to make them look pretty, and to exemplify what groups you're trying to show. Luckily, that's also easy in complex heat map. All you need to do is this function called heat map annotation, and then just put in the annotation table that you want to annotate on. So if you recall, this DESeq annote table is ignore that, I don't know what that is the annotation that we had before that we used for differential expression. And now our heat map plotting table looks like this, and you'll see that the column names here align up with the row names here. And so all in all, that means that it will be able to figure out that, okay, we're doing these samples and these samples, then these samples and these samples, and we're going to annotate these as treat and annotate these as control. So after you, but you don't need to do that much specification. All you need to do is run this one line. So this line here will create the annotation that you need. On its own, it doesn't look like anything. It's a complex heat map object, which is a bunch of stuff that you can look into more if you want later. It's not that important. But to actually include it onto our heat map, all you need to do is add on this one extra parameter, top annotation. When you do that and you draw it out, you will then get an annotation on your heat map. Now, I was alluding to before that in the end, we might not actually want to draw our entire heat map based off our top varied genes, because genes can vary differently and not necessarily because of the differences between our groups. Now, because this is a differential expression analysis lecture, we want to focus on the genes that were differentially expressed. So we're going to go back to our DESeq results table, and we're going to grab genes by whatever cutoffs you want, and then only show those genes. So what does that look like? Well, as with all my code, I start with the parameters that I'm going to pick our genes by up top with a p-value and log2 fold change. And then I'm going to select, not sure what that is. And then I'm going to select our genes of interest, GOI, using these cutoffs. So what this one line did here is select all those genes again by the log2 fold change and by the p-value. When you look at GOI, it's going to be a list of genes. Specifically, it's going to be a list of 592 genes. And now I'm going to make a heat map plotting table using just those genes. Now, with this heat map plotting table, it's going to look the same, but now it's going to have the genes in there I want. And I hope because that they're differentially expressed between the two groups, it's going to cluster out nicely this time. That would make sense. Now, the annotation this time, I also want to add on another piece of information. If you recall from last time, this data set uses both single end and paired end sequencing. This is our original annotation table. Now, I suspect that the single end and paired end sequencing might actually be altering our results, because you can tell here that treated 2 and 3 are paired end sequencing, while treated 1 is single read sequencing, and you can see that 2 and 3 look very similar to each other, and then 1 kind of looks a little different. So because I think that's a problem, I actually want to include that annotation in with our heat map. So this time when I make an annotation table, I'm going to grab both of these columns from here, running this line. You'll see that it will just look like this to grab these two columns. And then this time when I make my heat map annotation, I want to tell it what colors to use. Now, as I mentioned before, picking colors is a really tedious, but actually very, very important part of your visualizations. If your colors look bad, your whole thing is going to look bad, and you really want to spend a lot of time. Here, actually, I'm just picking some basic ones to get through the exercise. But in general, I would, again, as I mentioned before, I would not recommend to using the colors that come with just the base, red, blue, green, yellow. So here I'm creating a custom annotation for what I want my colors to be. You can dig a little more into this later, but it's pretty straightforward when you look at it, where basically I'm creating a list. And then for the condition column, condition, I am telling it that I want the treated ones to be red and the untreated ones to be blue. And then for the type column, I want the single end to be green and the paired end to be yellow. Pretty understandable code. So after I've done these two things, I'm going to create this custom color list. And this time when I have my heat map annotation, I'm going to put in the same annotation table I just made, and then this custom color annotation. So I do these three things. And then again, the actual HA top won't look like anything. You'll notice that there are two rows here for condition and type. But as far as these other things, those are aesthetics you'll have to tweak later. And generating the heat map is the same as before. And what this results in is, as we hoped, is a much prettier heat map. So here we have our new color annotations on top, and you can see that when we actually pre-choose the genes that are differentially expressed between our groups, when you allow it to cluster out based on what's there, you get this beautiful separation between your treated and untreated groups. Now, a question I get before is that, well, if you pre-choose your genes that are different, of course it's going to cluster out differently, right? So this isn't always the case. Just because genes come out in differential expression doesn't mean you're going to get this beautiful sorting. So this is still a valuable figure. This isn't a circular reasoning figure here. You will want to see what this looks like, because oftentimes when you pick out your differentially expressed genes, you may not get perfect sorting out like this. In fact, it may not sort out very well at all. So this figure will show that, one, you do have differentially expressed genes, and two, when you just let the math do its work, when you just let the clustering algorithm figure out what goes in what group based on the values in this table, it will be able to delineate out these groups, which in its essence, it's kind of a basic form of machine learning, where basically it's showing that using just this data here, it can separate out into an untreated and treated group. I will admit that I say here that I did add a couple other aesthetic tweaks just to make this prettier. There's a lot of things that you can do with heat map. In fact, let me just show you. If you press the tab button in R with a function, it'll show you all the things in. You can put in dozens and dozens and dozens of parameters in this. As I mentioned, it's really hard to make a really good heat map, and you may want to play with all these different things. But it only takes, these 2 lines to make a decent looking heat map. So it's as much or as little as you want. The last section in vignette is something that I already touched on before, which is the plea for good colors. I'm not going to say it again too much, but I'm just going to show you that here I showed what happens if you choose a different color palette. There's lots of color palettes out there. You can just Google for them. One of my favorites to choose from is just the Nature Publishing Group color palette. It looks good. It's generally black and white favorable, so it won't fade out with that. And yeah, I don't know. It's just a good idea to pay attention to your colors. So if I do all these things, you'll see that I will get a heat map out that will be nicely annotated, nicely colored. So, so here you have this whole vignette, which basically covers these 3 visualizations. Like I said, it was a little R heavy, but using this, you should be able to kind of plug and play whatever data you have, be it either count data or differential expression data, to generate figures that are good for explanation and also for publication. Using the remainder of the time, I'm going to talk a little bit about gene set enrichment analysis. So the end of differential expression analysis is a list of genes. And the question that always comes up next is, well, what do those genes do? A reasonable question. So how this question is most often answered is some degree of gene set enrichment analysis. So that term gets lumped into a couple different, a term is kind of an umbrella term for a couple different analyses. And there's really 2 big camps. There's one, which is essentially just an enrichment of the genes that belong to certain sets. And this is based off of what's basically just called a hypergeometric test. You're essentially just looking for an overrepresentation of genes belonging to an individual set. As in, if you have 50 genes you're interested in and 20 of them are all part of the coagulation pathway, well, that wouldn't happen by random chance. So there's a good chance that something's happening in the coagulation pathway. But there's a whole other set of analysis called actual gene set enrichment analysis. And that is a test that actually uses a specific metric of some sort to then rank your genes and then perform an enrichment test of some sort. So it's a little more complicated than that, so I'm not going to get into the details. What I am going to do is I'm going to show you a basic way to do your own gene set enrichment analysis. So if you're ever given a list of genes, you'll know something you can do to figure out what those genes actually do. So back to the slides, I wanted to show that using Broad's GSEA, you can do a pretty quick and dirty gene set enrichment analysis to give you some results. So using this link, we're just going to go to this website and we're going to investigate gene sets. Once you're here, you'll have to put in a list of genes and then you can investigate for whatever you want. So as I mentioned before, I downloaded that new DE-seq table that I put up in the Google Drive for everyone to use as a demo, because in here there are, this is a differential expression results from some other experiment, but it has a list of human genes that we can use. So if you're ever in this scenario, a good way to do this is to basically just take the table, sort it by an adjusted p-value or whatever you wanted to do, smallest to largest. I'm going to grab these genes. I'm also going to grab the log2 fold change. I'm going to go over here. I'm going to select all the genes that are, an adjusted p-value of 0.01, let's say. Mind, all this can be done in R too. I'm just showing for speed's sake what you can do here. And then I'm also going to apply a log2 fold change cutoff as well. So I'm actually only going to grab the genes that are also upregulated at least, let's say 2 in this gene set. So once I have this list of genes, interrogating what pathways they actually belong to is as easy as copying this and pasting it in here. Once it's in here, you just mark the gene sets you want to explore, and then you press compute. For those who have no idea where to start with this, I would recommend a couple, which is the hallmark gene sets. The hallmark gene sets are just 50 pathways of hallmark signatures. It's always a good place to start. The curated gene sets have a lot more in them, but these are the kind of things like KEGG and BioCarta that are curated sets for specific biological processes. And then Go terms are also useful ones. Go's a term other people might be familiar with, which is gene ontology, which is basically a systematic system for for breaking up different biological processes, cellular components, molecular functions, yada yada. I'm actually only going to do the hallmarks for now, though. So all you have to do, put the gene sets in, click hallmark gene sets, press the button, and here you go. It shows you your top gene sets enriched in this group. You'll see that it's this gene sets happens to be enriched for interferon genes and TNF alpha genes. You can dig into the results here, and you'll see what can make up these gene sets and so on and so forth. But there, quick and easy, not hard, and it's a way to just simply figure out what's actually in your genes. Now, I'm also going to show you how to do it in R quickly. In R, there's a number of functions of how to do this, but there's the one that I like best is called cluster profiler. So when we go into here, I'm going to start fresh, and I'm going to just read in that table that I was just looking at before. Here you can see that it's the exact same table I was looking before, and I'm going to grab genes of interest based off of the same parameters I had just said previously. So now I have these genes. Now, with these genes in R, we're going to use two libraries, Cluster Profiler and MSigDBR. MSigDBR is the MSigDB, just an R version that has been curated actually by Igor, who gave you a lecture previously. And then Cluster Profiler is a suite of tools used for gene set enrichment analysis. Now, I don't want to spend too much time on this because I'm really just kind of giving an intro as kind of a preview as to what you can do. And again, these vignettes are supposed to be used as helping tools that you can then plug in your own data into. But the short of it is that after we have loaded these two libraries, I'm going to set up the parameters we're going to use, which are, I'm using human genes. I'm going to pull up the hallmark gene set. If you recall, that was the same thing as here before, where I had the H gene set, the hallmark genes. And now I'm going to create this reference table where I'm going to pull out the human genes in the hallmark gene sets, this table right here. And I'm going to run this one function called enricher, which is going to take my genes of interest and then using this reference table, which just so you know, it looks like this, where basically it just tells the program what genes fall into what pathways. I'm going to leave me a p-value cutoff of 1 because basically I just want it to return to me everything. I don't want it to decide to get rid of things. I wanted to see what everything is and then go from there. So after I've just run these few lines here, I'm just going to run this function and I have my output, and then I'm going to see what it looks like. So it gives a bunch of stuff. And for our own reference, I'm just going to grab the very first line and I'm going to explain what's there. And then we're pretty much done. So we're almost done, just hanging in there for the last little bit. So as you can see, it's still the interferon gamma response, which was the first one before. You can see that it's just the ID of it. I am going to explain what these values are real quick because they're worth knowing, which is the gene ratio, 93/313. Where these numbers come from. 313 is the number of genes in your GOI that were anywhere in the universe you provided. And the universe you provided is based on the reference table that we just gave. So this reference table has these hallmark gene sets, which is 50 gene sets, and it's about, it's 50 sets, and in total, there are 4,000 genes within those gene sets. But of the GOI we actually gave that had 700 genes or so, only 300 of those genes are anywhere in the universe at all. So the other 400, we're just going to get rid of. We're not including in this calculation because it wouldn't be fair to test something that doesn't exist in this universe. So in this case, we're going to do, it says there's only 313 there. And of those 313, 93 are part of this gene set. And then in the background, 200 is the total size of the gene set. And as I said, there's 4,000 genes total in the universe as a whole. So you can see kind of basically these numbers, 93 over 300 versus 200 and over 4,000. Even just doing a little mental math, 1 over 3 versus 2 over 40. There's a significant over-enrichment for these genes of this pathway, hence the very, very high statistics. So that's basically what it means. I see we have a question, which I think is related to what I was just saying. Is the hallmark genes data set you are pulling what some gene enrichment software called background list of genes? So as I maybe kind of mentioned, but it's an important question, so I'll kind of answer it again, which is your background list of genes is what goes into this value of your hypergeometric test. Because in reality, you may actually want to alter what your universe of genes is to actually get the most accurate results. So for example, in this population, we started with something a long time ago, like 15,000 genes before doing all this filtering. So if we wanted to, for the most accurate statistics, we would use that original list of 15,000 and actually tell it that that's our universe. Don't infer the universe from our gene sets that we gave, but actually tell it, this is what our universe is. And you can see it, here's a parameter, you can actually say how many genes there are. So along those lines, that's actually the most accurate statistical calculation. Now, as far as this kind of quick exploration, the difference between 4,000 and 15,000 isn't that much considering how drastic these changes are. Now, if the change was much more subtle and you want to be precise, you would want to actually use the full universe of genes as you're doing this exploration and not just the genes that you have here. This is frankly a little bit where you get more into the art of bioinformatics as opposed to the science of it. People will do different things. They're all accepted answers. It's just important to back up whatever it is you do with some kind of line of reasoning. I think that's pretty much all I had for gene set enrichment analysis as well. And you can see here that I left a description of what each of these things are. Now, as I mentioned, the thing I didn't get into with gene set enrichment analysis is the specific kind of ranked test that more complicated tools do. It's a powerful method. It's very useful for pulling out subtle global changes. It's a little more nuanced, a little more finicky, which is why I didn't want to spend the time getting into here. But I wanted to show you just at least one quick way of doing via online and via R of how to do a gene set enrichment analysis. So with that, that's pretty much all I have. Everything you just saw, as I demonstrated here, is within this folder, within the Google Drive folder. So you can pull everything from here and do everything that here from home. I really try my best to make the vignettes as plug and play as possible. If you have any questions, of course, you can ask me and I'd be more than happy to help. And with that, I think I'm going to wrap up. I'm going to stick around for questions as usual, but I think that's all I have to display. So thank you very much for coming. This was the last lecture of the year, and it's been really a pleasure teaching you guys, and I hope you all learned something during this. Thank you.